Yeah, thank you so much for the kind invitation. It's a great pleasure to be, let's say, although virtually, to be with you today. And I'm very delighted to share some unpublished data from recent work that I've been conducting on a novel NAD precursor, and we will touch upon NAD metabolism a little bit, which is called Trigonelline, with muscle longevity benefits across species. Just as a small disclaimer, I mean, most of the work that I'm presenting today was actually conducted when I was leading my research team in Nestlé Research in Switzerland. And then I'm sort of finishing it off here since I joined the Healthy Longevity program in Singapore under Brian Kennedy. So with this in mind, the focus clearly of the, let's say, of my research interest, where I'm referring to in terms of the data you will see today, is about muscle aging and sarcopenia. And that's because when I was in the research team in Switzerland, one of our interests was to find nutritional solutions, or anyways bioactives, that would allow us to ameliorate muscle aging and sarcopenia. And just to give you an idea of how the process of muscle function goes and declines as we age, here on this sort of just virtual or qualitative plot, you can see on the y-axis muscle mass and strength, which change, of course, through age. And we can separate, let's say, if you want, muscle health across 3 stages of life: early life, adult life, and older life. You may be aware that, let's say, muscle function generally peaks in our early life until, let's say, perhaps our early 30s. And then the idea is for us to stay as functional as possible for as long as we can be as we age. And of course, we are all different. I mean, there is all kinds of factors that come into play that regulate our muscle functionality, strength, and so forth. And there is a range of strength which allows us to remain functional, but there is also people which unfortunately are, they have a steeper decline in their muscle health. And when this goes below a certain threshold of physical performance, we reach disability, therefore muscle sarcopenia. In quantitative terms, there are consortia throughout the world. Here's the European consortium and the Asian consortium for essentially sarcopenia definition, which have highlighted or agreed on 3 main muscle health clinical parameters: gait speed, grip strength, and muscle mass. And pretty much the threshold values that they use are quite comparable, I would say. So this is important to keep in mind, because also in our observational studies, we will refer to these 3 clinical outcomes. However, from a biological perspective, when we go in the molecular pathways that affect muscle aging and sarcopenia, it's important to notice that some of these are essentially shared with the hallmarks of aging that have been described abundantly in literature so far. And for muscle aging and sarcopenia particularly, some of the hallmarks that are very critical to this process are the dysfunction of satellite cells, increased protein catabolism, and therefore also decreased protein synthesis, which is key in muscle, increased fibrotic infiltration, or increased inflammation. But since muscle are very metabolically active and they contain a lot of mitochondria, which generate the energy for, let's say, for our regular function, 2 of the key hallmarks in muscle dysfunction are dysfunction of mitochondria and therefore also increased ROS production. When we go to, let's say, real life conditions, this is an observational study that was carried by my team before I joined essentially the research team in Switzerland, which was published in 2019. What they did, they tried to look indeed at what would be the biological hallmarks of sarcopenia, looking at different cohorts in humans and comparing control patients versus sarcopenic patients. This in particular, the data will show, is referring to a sarcopenic cohort from Singapore, which was assessed by the 3 parameters that I just mentioned before. So appendicular lean mass, grip strength, and gait speed. So when patients were essentially separated between control and sarcopenic based on these parameters, muscle biopsies and serum were collected. And from this study in 2019, what was performed was essentially only tissue analysis, so from the muscle biopsies. And really, in pretty much one sort of screenshot, what emerged is that if you do, for instance, transcriptomic from muscles, most of the downregulated pathways here seen in blue from sarcopenic patients are essentially referring to mitochondrial gene expression. And it turns out that, in fact, in these patients, as I mentioned you before in the cartoon, but now with also real data from this observational study, mitochondria gene expression and function are strongly downregulated. Also, what emerges is that if we perform metabolomics analysis from these tissues and we look at things like, for instance, NAD levels, which are a key, it's a key cofactor, which I will explain in a bit, but it's also related to mitochondria and muscle health. This also turned out to be significantly downregulated in sarcopenic patients, and across the two groups was actually positively correlated with grip strength, lean mass, and with gait speed. Therefore, in a way, saying that having more NAD levels in muscle could be linked to better muscle health overall. Now, just to, as I mentioned, since NAD is a very key cofactor, it's basically not to go into the details of NAD metabolism too much, but perhaps you may be aware, and also looking at this cartoon from this nice review from David Sinclair, that NAD has been linked essentially to almost most of the hallmarks of aging and key cellular homeostasis processes, such as indeed mitochondrial function, redox reactions, inflammation, DNA repair, and so forth. And also there is now a plethora of preclinical studies and also now some ongoing clinical trials that are trying to go in this direction, showing that if you're able to maintain NAD levels through, for instance, exercise or diet regimens, or even using NAD boosters, such as in the case of Trigonelline, which I'll show you in a moment, this is actually linked to all kinds of benefits for pretty much any type of tissue or organ you could think of, including, of course, muscle benefits. Now, having analyzed already the muscle biopsies from these patients and having found a link between mitochondria and NAD metabolism in these patients, we wanted to look at systemic factors in the serum samples from the same cohort to look at either new biomarkers associated with sarcopenia or muscle decline, or even by sort of causal factors that could be linked with somehow with the onset of the disease, if you want. When we performed a metabolomic analysis of different vitamins and metabolites of relevance, let's say for the aging and for muscle, we didn't actually see too much, except one particular metabolite, indeed trigonelline, which was significantly reduced in the serum of patients with sarcopenia. Interestingly enough, serum trigonelline levels correlated again now positively in the cohort with again the three clinical parameters of muscle health. And this actually prompted us to look at its potential link with NAD and mitochondria. And in fact, it turns out that the NAD levels in muscle that I presented in the slide before, they were also partially associated with the serum trigonelline levels. And when we look now at the transcriptomic analysis again, and we try to enrich, we look at the positively enriched pathways that are linked to higher levels of trigonelline in serum, the most enriched pathway in this case was again oxidative phosphorylation. In this case, now it was a positive enrichment, and therefore, as you can see here, most of the oxidative phosphorylation genes are indeed upregulated when there is high trigonelline in serum. Therefore, we can sort of, our concept from these slides is that trigonelline in serum correlates with NAD and mitochondrial gene expression in skeletal muscle. Now, this, of course, brought us to the question of how does trigonelline look like and what does it do? So when we look at the structure of trigonelline here, it turns out that trigonelline is a methylated form of nicotinic acid that you can see here on the right. And we all know that nicotinic acid is a very well established NAD precursor indeed. Trigonelline itself is actually, it's been known for a while already since the 19th century for its anti-diabetic effects, although the mechanism of action is still under investigation. There is also a lot of preclinical evidence showing that, for instance, trigonelline decreases plasma and liver cholesterol in liver pathology conditions, and also can modulate glucose transport both in rodents and in human cells. However, what's important to keep in mind is that trigonelline is actually largely present in plants and produced in plants, and we do not know how much is actually made or at least could be found in mammalians yet. And that's because plants usually what they do, they produce a lot of nicotinic acid from the NAD cycle, and then in some species, especially green coffee beans, fenugreek seeds, for instance, and some type of foods that we ingest through the diet, they do methylate nicotinic acid to produce trigonelline. So with this in mind and the observations that we had from our human cohorts, our question became whether trigonelline could act as an NAD precursor in animal species and impact longevity and muscle health. So before we dig into the data, I want to bring you also a little bit up on the context of the different NAD pathways that lead to the production of NAD in our bodies. So there are at least three types of biosynthesis pathways. The price handler pathway, which makes use indeed usually of nicotinic acid coming from the diet. And in this case, we, being trigonelline, a sort of methylated form of nicotinic acid, we speculated that it would be through the price handler pathway. Then there is the de novo synthesis pathway, which usually makes use of tryptophan degradation. But then also there's the salvage pathway, which is perhaps the most exploited preclinically and also now object of interest from the clinical investigation perspective, since usually makes use of very strong NAD precursors such as nicotinamide from the diet or nicotinamide riboside or NNM, which are also available. All of these are available as supplements nowadays, and people are testing these in the context of clinical investigation. And they are usually very potent because they're more directly linked to the production of NAD through either two or even one enzymatic conversion. So with this in mind, we decided to test trigonelline in different models, and we started with human myotubes, so human primary skeletal myotubes, where we started providing trigonelline at different doses, and we indeed assessed, compared to the control here in black, that trigonelline can increase NAD levels. Same goes when you use primary human myotubes, even from sarcopenic cells. Although these cells come from patients, they're still able to respond to NAD interventions such as trigonelline. And that's very important if in the perspective of wanting to use this for clinical applications. We next moved to an in vivo system, which would be short-lived in order to assess the effects of aging. And in the biology of aging, we often use nematodes, C. elegans, in order to study aging. So what you can see here is that if we look at NAD levels in young worms or in old worms, and the only difference here is about 8 days of adulthood. So by day 8, these worms already show decline in NAD levels. When these worms are treated for about, indeed, 1 week with trigonelline, we actually maintain the NAD levels to a more youthful state. And when we gavage, so we make mice ingest trigonelline, and then we look, after 2 hours, we look at the tissues and we measure NAD. We can actually indeed prove that also in mammalians, in different tissues, simple ingestion of trigonelline can lead to increase of NAD levels across basically all the tissues we observed. Next, we wanted to actually investigate the pathway and verify that indeed our hypothesis that trigonelline would use the price handler pathway would be correct. So in this simplified cartoon here, I'm showing you the price handler pathway again on the left side and the salvage pathway on the right side. What's key to keep in mind is that nicotinic acid relies on this enzyme called MPRT for its conversion into the NAD pathway. And this enzyme can be blocked using, for instance, a compound. Instead, it makes use of the NMPT enzyme to recycle essentially NAM from the NAD degradation and to put it back into the NAD cycle. And it can be very precisely inhibited through this drug called FK866. And then you can make use of the different precursors in order to assess the pathway relevance. So for instance, for nicotinamide riboside is downstream of these two pathways, so it won't be affected by using these inhibitors. And that's what we can see here in this first experiment. So we gave to primary myotubes trigonelline and NR, and we could assess that both increase NAD levels compared to control cells. However, when we now use the 2-hydroxynicotinic acid to block the price handler pathway, nicotinamide riboside just works fine because it's downstream of all of this. However, trigonelline effects are completely lost due to inhibition of the price handler pathway. And now we did also a complementary approach where we did the same setup, but where we use the FK866 compound to block the salvage in these cells. And what's very important and nice to see here is that when you use this compound, you're basically blocking a lot the bulk production of NAD in these cells. Therefore, you get a very strong depletion of NAD levels when you treat with this drug. However, very importantly, because of NR, which is downstream of this, and because trigonelline relies on this pathway, we could actually completely rescue the NAD levels with trigonelline and even further boost them with NR. However, when we now combine the two inhibitors, NR still works, while trigonelline effects are again now completely lost, again confirming the reliance on the MPRT. And with this in mind, so keeping in mind the cartoon that you can see here and the effects on NAD that I just showed you before, we look now at the effects of NAD depletion on mitochondrial function. sort of to link again NAD alterations with mitochondrial health. You can see here which mirrors sort of what we see here with the NAD levels. You can see that in cells which are depleted of NAD, mitochondrial membrane potential declines, but is fully rescued by both trigonelline and and NR in the condition of FK66, so salvage inhibition. But when we again block now the two pathways, only NR can rescue mitochondrial membrane potential, while trigonelline fails to do so to a large extent. And when we go even deeper in mitochondrial function and we measure the oxygen consumption rate, in particular, in this case, we are showing the maximal respiration here, which is the maximal ability of the mitochondria to respire once you uncouple essentially the mitochondrial membrane potential with the FCCP. We can see that again, the maximal respiration is affected by the depletion of NAD and is completely rescued with the two compounds, but not by trigonelline when you inhibit the Price-Handler pathway. So having confirmed this in cells, we reverted back to a short-lived model of aging, so in this case, C. elegans, and we performed sort of the same study by using now genetic epistasis to block the MPRT ortholog in worms through a knockdown experiment and see whether we would lose the effects of trigonelline on nicotinic acid, on NAD, and also essentially on lifespan, because in nematodes, there is a lot of studies that have actually linked essentially increased NAD levels to extension of lifespan, or anyways, maintenance of a lifespan in that case. So that's what we see here. When we perform lifespan experiments in C. elegans, you can see in black, this is the control condition. When we use nicotinic acid or trigonelline, we get actually comparable lifespan extensions with both compounds. However, when we block now MPRT through a genetic repression, so with a knockdown approach, we don't see the compounds being able to extend lifespan anymore. So this sort of fits a little bit with the cellular experiments, but now looking at phenotypes, or whole body phenotypes in this case. And when we move to health span and looking particularly in muscle health in the nematode and treating the worms again from day one of adulthood and then following them as they age, you can see parameters of muscle fiber integrity in these worms. For instance, the muscle fiber integrity is largely lost in aging. This is at 11 days of aging compared to day one. You can see the loss of integrity. However, when we treat the worms chronically for about 11 days with trigonelline, we largely preserve the muscle structure, so we ameliorate the muscle loss phenotype. And this is mirrored by things such as scoring for paralysis, which it's basically as the worms age, they get more and more paralyzed because they lose mobility. However, with trigonelline, we are able to significantly reduce so. And this is mirrored on the other end by an increase in spontaneous mobility, even at old age in these worms. So these are day 11 worms, which are still significantly moving more than the control worms. What happens now when we translate these findings all the way back to mammalians, and in this case, to aged mice? So we did a study in collaboration with the Gordon Lynch lab in Melbourne, where we basically took old mice about 20 months old, which is, let's say, quite already a good aging, let's say, moment for the mice, where the mice are properly aged. And when we treated them for about 12 weeks, so 3 months, with trigonelline at this dosage in the food pellets. And then we had an aged control diet group. At the end of the study, we performed things such as, for instance, grip strength or in situ muscle force. In situ muscle force means that you take a muscle in particular, such as the tibialis anterioris in our case, and you connect it with electrodes, and then you stimulate the muscle to contract at its max. And then you stimulate through several contractions, you induce the muscle to basically get fatigued due to the repeated contractions. So at the end of the study, we looked at things such as lean mass, grip strength, muscle fatigue. And then what we observed is that lean mass actually in peak force, which is really the maximal, first maximal contraction that you induce in the, for instance, in the TA muscle in this case, were not affected. However, when we now induce muscle fatigue through repeated contractions in the tibialis anterioris in this case, and we look at a comparison between young aged and age-treated mice, you can see that the young mice here, they do get fatigued when you perform multiple contractions of the muscle. However, then the muscles, the phenotype sort of stabilizes. When the muscles are old, so when you are aging, essentially your muscles fatigue much quicker. So there's a steeper decline in muscle, let's say, force, which is an indication of fatigue. But with the trigonelline treated group, we're actually sitting somewhere in the middle. So we get a partial rescue of the muscle fatigue. And this is also mirrored in vivo before we actually sacrifice the mice by a small but significant increase in grip strength in the trigonelline treated aged mice. Therefore, trigonelline, with these results, we can conclude that trigonelline sustains force during high-intensity contraction and increases grip strength in aged rodents. And to conclude, in terms of take-home message and remaining questions, what we discovered is trigonelline levels decline in a human sarcopenic cohort and associated with mitochondrial function. We have shown that trigonelline is a bona fide NAD precursor across species and using or mainly relying on the Price-Sandler pathway. It ameliorates muscle and mobility decline in nematodes, but also sustains contraction in aged rodents. And then there are remaining questions, of course, that will be addressed in further studies, which concern the demethylation process of trigonelline, which in mammals remain to be sort of properly characterized. And of course, the clinical translation of trigonelline for muscle health in humans. It's something that needs, of course, to be done following these first preclinical studies. And then any additional benefits and mechanisms of action, even maybe related to NAD that could come from using trigonelline, which of course require further studies as well. And with this, I want to conclude by acknowledging the NUS and the Center for Healthy Longevity, who welcomed me about half a year ago now. So I want to thank Professor Kennedy and Andrea Meyer, my department head, Professor Wenschen, and all the members from the Center for Healthy Longevity. My ex-team and Nestlé Research, with which we are still actively collaborating on finishing this study and further studies, my collaborators in Melbourne, and also my ex-business partners from Nestlé Sciences, with which we conducted some of the studies together. Thank you, and I'm open for any question if there is.